Welcome to the Biology 182 lecture series on classification. This video is about molecular clocks. At the end of this video, students should understand how divergence and evolutionary relationships among species are determined. This graphic shows the divergence of organisms from a common ancestor using a gene. The molecular clock studies the DNA or genes of an organism to measure the evolutionary time between divergence of species. So the two organisms' relatedness we're measuring in distance of gene change that we correlate to evolutionary time. There will be one common ancestor and two taxa arise that diverge, in this first case, 25 million years later. Then there's another separation about 50 million years later, and you can see that the accumulation of DNA mutation occurs at about 25 million years and at about 50 million years. These known divergences are often used to determine evolutionary time in conjunction with the geologic record and the fossil record. In order to understand the molecular clock theory, we need to understand the neutral theory of evolution. Muthu Kimura hypothesized that changes in genes or proteins that do not confer a selective advantage, so there's no disadvantage or advantage to the mutation arising, in order to enhance or create a detriment in survival, would accumulate at a steady rate. So neutral changes in genes or proteins accumulate at a steady rate. Well, we have come to learn that this actually varies by organism. So it's not quite as simple as he originally had proposed. Neutral mutations follow a few different ideas of the neutral theory and the nearly neutral theory. But before that, we had the selection theory. And the selection theory said that all mutations arise and they affect fitness in one way or another. They're either advantageous or deleterious, and deleterious mutations would rapidly be moved out of the population because they're hurting the fitness of an organism, where advantageous would surely go toward fixation or a more universal acceptance of the genome because they'd be advantageous. The neutral theory says that neutral mutations arise and are accumulated at a faster rate than either deleterious or advantageous. And this depiction shows that the deleterious still arise, but super rarely the advantageous. And the deleterious are rapidly removed again from the population, but the neutral are going to stay. It's not going to be noticeable, the change, because it won't affect fitness or selection. The nearly neutral theory says that the majority are deleterious and removed, are nearly neutral or neutral, and the remaining are the advantageous rare. But for the most part, these nearly neutral and neutral mutations that arise, they confer really no selective advantage or detriment, and they are produced at a faster evolutionary rate of change than other mutations are, but nearly neutral because this is dependent entirely on the size of the population. Small populations, you can't have a nearly neutral mutation. It is going to have an effect or an impact because the population's so small. Where in a huge population, it would be nearly unnoticed change. At this point, these changes in our DNA or our genes are causing or creating what are known as pseudogenes. Pseudogenes were at one time a gene that worked, right? And it coded or was translated into something with a purpose. So these pseudogenes may have now have a section missing from the genome that now it's missing its promoter sequence, or now it doesn't have its start codon. Maybe it's a frame shift mutation, so it now is a completely different protein that it codes for, or it prematurely stops. Maybe it's missing the intron, so exons are smashed together in the wrong place, and now it's going to excise the wrong part of the gene. Or part of it was deleted that was needed. These mutations are causing a gene to become a non-coding sequence of DNA, which we once thought was just all junk DNA. We had no idea what the purpose was, so we referred to it as junk DNA. These are also known as introns. They become pieces that are non-coding or unfunctional. This functionless evolutionary relic, I mean, I'm thinking we could call them vestigial DNA, right? They're no longer functioning. They have no purpose anymore in our body. But maybe the accumulation of mutations in these pseudogenes will show us the rate at which the organisms are diverging or evolving. Mutations arise at one point, right, in our DNA. And so they are called single nucleotide polymorphisms. Unless a chunk is lopped off somehow, there is a single nucleotide or one base change in our DNA that arose from the mutation. These changes can cause various effects. We learned in 181 they can cause a missense mutation, where now this one base codes for a different base, the third wobble in a codon that now reads for a different amino acid. So the entire protein is the same except one amino acid is different, and that could change the protein. It could change its shape, change its function, and it may not be quite as noticeable depending on which amino acid it codes for. We've learned they can become nonsense mutations, which just stop early the translation of this gene into the protein. Or it could become a silent mutation that we don't even know exists because our DNA is our genetic code is so redundant. There's 64 codon possibilities, but only 20 amino acids coded for. So oftentimes they're silent. We don't even know that they're there. Additionally, they could be a frame shift mutation that arise from like the addition of a base that shifts the reading frame downstream, and now it's a completely different protein, or a deletion that shifts the frame a different direction, creating a whole new reading frame downstream. So all of these mutations are at one single point, the SNP or the SNP, right? Most of them are neutral. What this means is the redundancy is a huge factor, or it happens in an intron, right? And so we don't even notice because it wasn't coding for anything anyway. Or it confers no survival advantage or no survival disadvantage. And because of that, maybe it's something like, oh, this wild tiger now has one blue eye and one brown eye instead of two brown eyes. Do we think he now is going to be able to catch more food? Probably not, right? There's not really a survival advantage to that. Or this domesticated house cat now has a pattern of black and white instead of just being black. Well, I'm thinking that's not going to confer any kind of survival advantage either for this domesticated cat, right? So there are some instances that there is no survival impact. And because of that, we refer to them as neutral mutations. Well, molecular clocks capitalize on the neutral mutations, and we look at how we can convert these changes or deviation in sequence of our genome to time, how the organisms or species have diverged between their lineages over time. But we also have to make a correlation with biogeographical and fossil record data because nothing is perfect at this point, right? The biogeographical data we look at is how the Earth changes and how the continents shift and change and how the oceans shift and change based on the continental and tectonic plate shifting, right? How these organisms are affected over time from these changes can also be seen by the Earth shifting or changing. We also look at the fossil data in the fossil record, but this is not always clear either. We can sometimes see changes in the fossil record that aren't reflected for millions of years, and they may have changed sooner than that. So in this example, we're looking at the large ribosomal RNA subunit. So the ribosomal RNA of the large subunit has changed so much over billions of years, from when life first originated to breaking off into bacteria and archaea, and finally into this eukaryotic branch that we see our life arise from, right? All of these changes happen, and these changes accumulate over incredibly large amounts of geological time. They are not quick. Changing of the RNA of ribosomes is so slow that this mechanism to analyze the molecular clocks of these neutral mutations in the ribosomal subunits, or large or small subunits, is so slow that it's awesome for depicting the time frame of changes in domain or changes in kingdom, right? Supergroups. But that's not so great at determining the changes among families or genus, right? Because they occur at a faster rate. And so now we're looking at different ways to address these changes. And so though molecular clocks are not perfect, the fossil record and biogeography are not perfect, because none of us was actually there hundreds of millions of years ago to see when these things arose. The best of our ability at this point is to combine these ideas with molecular clocks and the fossil record and biogeography to bring together a whole picture. And so one of the things that they look at is rocks and clocks, right? And so the rocks are the biogeography and the fossil record, and the clocks are our molecular data and how these neutral mutations arise and the rate at which they come. And they do not agree. Right? So the clocks point the evolution of birds to have arisen about 75 million to 160 million years ago, where the rocks tell us that we have fossils of birds from about 62 to 66 million years. But the DNA evidence or the RNA evidence cannot be discounted, because at what point were the fossils first becoming fossilized 60 million years ago for these birds? Was it the very first organism? We're not sure. Did the divergence of molecular accumulation of these neutral changes happen at that very first moment of change, or did it give rise to change over time? How these deviations in time spans is still not perfect is going to mean that the usage of all of these things together is going to come up with the best picture possible. But one of the nice things about this is we can see the changes follow trends and patterns together, that the trends on when these things arise will follow the same pattern. Even though it's a few million years different, it will still follow the same patterns between the clocks and rocks. And so one of the things that we've come to realize is that species are so different. And as these mutations arise, the rates of them arising is totally separate. You can't compare one to another. Even in really closely related species, you can't compare. They still diverge quite a bit. And so one of the things that's believed in rocks with fossil record is that after extinction, a huge diversity burst will come, and we see tons of new fossils, right? Well, new species come in addition to new fossils with new neutral accumulation of mutation. And so there's a faster rate of neutral accumulation and mutation, as well as a diversity we see in the fossil record. But one of the things that we do see is that mitochondrial DNA is going to be fast changing, where those ribosomal RNA changes that we were looking at were very slow moving. So those were good for, like I said, the domains and the super kingdoms or supergroups, right? But they're not so great at genus or phylum or family order, right? These we want to look at mitochondrial DNA changes, because the cytochrome B is very fast at accumulating neutral mutations across the board. However, by very fast, we're still talking about a 1% accumulation of change in a DNA or gene over 1 million years. It's still a very long time. It's not like next week we're going to see an evolutionary change from humans to start branching off into a new species, right? But these million years, you will see a 1% change. This evolutionary rate occurs about 1% every million years in birds. Well, in some birds, it actually occurs about 8% per million years. And so the rule of thumb has always been to this point that 2%. We're at a 2% divergence between species per million years. But like I said, some are slower in some birds, some are way faster in some birds. So nothing is consistent because every species is different. So in reality, nothing is perfect. But for our best data at this point, we look at the molecular data in MT or mitochondrial DNA, and we use the support of the fossil record and biogeography to help create that bigger picture of evolution. Students should be able to understand molecular clocks and describe the relationship between molecular clocks, the fossil record, and biogeography.